Hey everybody, welcome to our first online immuno lecture. The plan for today is to talk about major histocompatibility complex. I know we've spoken a lot about MHC already as far as what its role is in the immune system. But the goal of today is really to dig a little bit deeper into the genetics of MHC and understand the clinical importance and applications of MHC to you as a lab tech. So before we jump into the genetics, let's just quickly recap what we already know about MHC. First, we know it has a crucial role in the activation of the adaptive immune system. Antigen-presenting phagocytes such as macrophages, dendritic cells, B cells will ingest foreign particles and destroy them, after which they'll present a piece of it on MHC2 for T helper cells to recognize. The end result of that is antibody production. MHC2 we know is present primarily on antigen-presenting phagocytes. Now, in cases where, let's say, viruses invade a cell before phagocytes can get to them, the cell will take pieces of it and present on MHC1 instead, assuming, of course, the virus hasn't already compromised the cell's ability to produce MHC1. So it'll do this for cytotoxic T cells to recognize and destroy. And we know that MHC1 is present on all nucleated cells in the body, or almost all. So they're named MHC because it's these molecules that determine whether a transplanted tissue will be accepted or rejected by the host. Historically, that's why it's named that way. Histo for tissue and compatibility, well, that speaks for itself. So hence the term histocompatibility. So we'll see later that a person's MHC genetic profile is highly, highly unique, almost like a fingerprint, and is very difficult to find a perfect match between donor and recipient, unless it's from within the immediate family. The term MHC is also used interchangeably with HLA. So when determining tissue or organ compatibility, we perform a procedure known as tissue typing, which we'll see in future lectures. But all this procedure does is it aims to identify the different types of MHC antigens present on the surface of a patient's white cells, hence the term human leukocyte antigen, or HLA. So the closer the donor and recipient's tissue type or white cell MHC antigens match each other, the lower the likelihood of organ rejection. And finally, how strong a person's immune response versus someone else's against a particular foreign substance is largely dependent on the exact MHC profile a person inherits. And so as far as inheritance goes, class 1 and 2 are both coded for at different locations on the same chromosome. So let's talk about the structure of the MHC molecule for a second. And what you're seeing here on the screen is class 1 and class 2. They look a little bit different in terms of structure, but this is what they actually look like on the surface of the cell. You can see that class 1 over here is connected to the cell membrane at one point here, right here. With a so-called tail, and consists of an alpha and beta chain. Okay, the alpha is actually folded into 3 sections, alpha 1, 2, and 3. And the beta chain here is kind of by itself. It looks like it's free floating. It's not really, it's just weakly connected to the rest of the structure. And it doesn't really serve any known purpose at this point, except to say that it probably serves as a structural support for the rest of the molecule. But beyond that, it's not believed that there's any real function to it, except for structural support. Class 2 molecules are connected to the cell membrane in 2 spots. You can see right there. And they have alpha and beta chains. But the main difference between this and class 1 is that they're all connected to each other. So you can see alpha 1, alpha 2, beta 1, beta 2, but they're all connected. So what I want you to notice here is the peptide binding cleft. Okay, the MHC molecules are structured in such a way so as to create what's known as a cleft or a crevice, and it's inside that crevice that pieces of antigen peptide are held for T cells to recognize. So, like I said, MHC1 and 2 are coded for on chromosome 6, with the exception of the beta chain, which in class 1, which is actually coded for separately on chromosome 15, but we'll be focusing on chromosome 6. Okay, so some inheritance basics. In humans, each cell normally contains 23 pairs of chromosomes for a total of 46. Half of that, so 23, comes from mom, and the other 23 you inherit from dad. So each chromosome contains your genetic material in the form of multiple genes, okay, at different locations on the chromosome that are responsible for coding a variety of things, ranging from hair and eye color to the most sophisticated of functions like enzymes responsible for catalyzing essential biochemical reactions and everything in between. So with respect to the type of MHC profile we inherit, the work is done on chromosome 6, like I mentioned. So what we're seeing here is a single chromosome 6, and the MHC molecules are coded for in this location in the short arm of the chromosome. Okay, so this is kind of a blown-up view. And so we can see all the different genes. Okay, so that's what we'll talk about. MHC class 1, which you can see right here, it's coded for in these locations here. There's 3 different locations, okay? Locus A, that's the terminology, a locus for a location. So there's a gene here, locus A, locus B, and locus C, okay? Each of these locations codes for different versions of the MHC1 molecule, each of which is capable of displaying a variety of antigenic peptides. Okay, so you got a bunch of different MHC1 molecules that are produced here, a bunch here, a bunch here. Okay, each of which are capable of producing or displaying a variety of different types of antigen. MHC class 2, same kind of principle. It's just located. The genes are all located in a different region of the chromosome called the D region. So those regions are specifically called DR, DQ, and DP, okay? And it's the same thing here. Each location codes for different versions of MHC class 2. All right, so you'll also notice that between class 1 and class 2, okay, you've got class 3. We're not really going to be spending any time talking about class 3. I just want you to know that it exists. It codes for different certain complement proteins and cytokine production. Okay, but we're not really going to focus on that. I just want you to know that it's there, that it exists, and it codes for things. It codes for other things. So that's it. As far as working knowledge about class 3, that's all you need to know. Our focus is really going to be on class 1 and class 2. So very important to note, to take note, is that the MHC molecules produced are co-dominant, okay? Co-dominant expression, meaning that whatever is produced by these genes, okay, they're all equally expressed. Those MHC antigens are all equally expressed on the surface of the cell, okay? There's not a preference for class 1 over class 2. One doesn't dominate over another. They're all equally expressed, hence the term co-dominant expression. Okay, it isn't like the hair color example we talked about, where the child inherits, say, a dark hair gene from dad and a blonde hair gene from mom. In that case, usually the dark colors dominate and will be expressed, despite the fact that the child does also have a gene for light color. So in the case, that's not the case for MHC. Everything produced is equally expressed on the cells. So we mentioned in the beginning that the MHC profile a person inherits is very unique and almost like a fingerprint, almost it's virtually impossible to duplicate it in another individual outside the immediate family. So the tables in this slide tell us why they're so unique. So recall if let's talk about class 1 as an example. We'll just stick with that. So recall that we've got 3 locations per chromosome 6 where MHC1 is coded, locus A, B, and C, right? So since we all have a pair of chromosome 6s, this means we'll each end up with 6 different varieties of MHC1 antigen being expressed on our cells. So look at the table on the top left right here, okay? It's telling us that as a human species, locus A has been known to produce 28 types of class 1 antigen. The 6 any individual ends up with essentially comes from that pool of 28, okay? The antigens have a numeric nomenclature, okay, shown in the table over here. Okay, they're not necessarily numbered 1 to 28. The specific numbers are listed here. So it can be called HLA A1, HLA A9, all the way up to HLA A80. Okay, it's not necessarily HLA 1 to 28. Okay, so there's a nomenclature for it. So 28 different types of antigens, but further adding to the variability is the number of alleles associated with the antigens for that locus. So you can see alleles right here. Okay, 3,356 different alleles. So what's an allele? It's a slight difference in the product that's made, but not different enough that you need to reclassify it as something else. You can think of it like an antibody allotype. IgG has 4 different versions of itself, but and they each have slightly different functions, but they're all still essentially IgG. So for humans, locus A here has 28 main types of class 1 antigen, with each having several possible variations or alleles, totaling 3,356 for that locus. So HLA-A1, for example, may have 200 different versions of itself. HLA-A2 may have another 145 of itself, so on and so forth, for a total of 3,356 variations or alleles for that locus. Okay, so when you consider the thousands of different alleles across all loci, you can begin to see that the chances of any one individual ending up with exactly the same MHC profile or tissue type as someone else is next to nil. Okay, so it's important to recognize that much like the IgG example, each MHC allele has slightly different functional capabilities in the immune system. So we know that the main role of the MHC molecule is to be able to present non-self antigens for recognition by T cells, right? But the fact is that not all MHC molecules are equally talented at performing that task. So what one version can't do well, some other version might do better. It comes down to this: the greater the diversity in the MHC gene pool, the more diverse the immune response a species can mount against an infinite number of pathogens. Okay, this extremely high degree of variation is known as polymorphism. It's in your notes. Okay, so just take note again that this high degree of variation is known as polymorphism. Okay, the MHC gene pool is highly polymorphic. And in fact, the survival of our species has been in large part due to this polymorphism. So the table on the bottom, okay, is telling us that depending on the exact alleles you inherit, you may be more susceptible to certain diseases. It doesn't mean you develop the disease, it just means there's a greater chance that you could. So let's go through the examples that are seen here. If you've got HLA-B27, okay, there's a very strong association with the development of this disease here called ankylosing spondylitis. which is basically a severe inflammation of the vertebrae of the spine. Okay. Celiac disease, something that's more and more common. In fact, my wife has got celiac disease. My eldest daughter has celiac disease, and to develop that disease, there's a strong association with having these HLA alleles, so DQ2 and DQ8. So these are class 2 MHC molecules, okay? And there's a very strong association between DQ2 and the development of celiac disease, less so for DQ8, okay? So when we did our genetic testing, I've got DQ2, my wife has got DQ8, okay? And it turned out that my wife and my eldest daughter developed celiac disease. I do not have celiac disease. Okay, so it doesn't mean you develop the disease, it just means there is a stronger possibility that you could, okay? Also, type 1 diabetes is highly associated with those same HLA alleles, DQ2 and DQ8, okay? So it's just something to keep in mind. Okay, so let's get into an example of MHC inheritance. So on the top left, right here, okay, let's say this is dad's MHC profile. The right here would be mom's, let's say, okay? Each color strip that you see represents a single chromosome, okay? So we all have a pair of chromosomes. So dad's chromosomes represented by the red strip here, A, and the blue strip, B, okay? And mom's represented by the green strip, C, and the yellow strip, D, okay? So a pair of chromosomes for dad, a pair of chromosomes for mom, okay? On here you have the alleles that they've, the HLA alleles that they've, that are on each chromosome. So dad has A1, he has CW7. So this is just, they added the W for the C locus just to not confuse it with complement proteins. Since class 3 is on the same chromosome and it codes for certain complement proteins, they just added the W just to distinguish it from complement, okay? So dad's got A1, he's got C7, he's got B8, DR3, DQ2, okay, on one chromosome. On the other, this is what he's got. Mom's got this on each chromosome, okay? So the idea here is to determine the different possibilities as far as what their offspring might inherit, okay? All the possible combinations are below here, okay? And based on the fact that half of dad's genetic material gets passed on and the other half comes from mom. We just don't know which half from each parent gets passed on. So this is why the answer is formed in terms of probabilities, okay? So just run through all the possibilities here, okay? So let's say dad passes on A here to his child, okay? So if dad passes on A, that's this half from dad, the other half has got to come from mom. So it's possible that mom will pass on C, okay? So one possible combination is AC for the offspring, okay? Another possibility is dad may pass on A right here, okay? But instead of mom passing on her C, she'll pass on D, okay? So that's a combination there. Dad may not pass on red at all. He may pass on B instead, okay? So B is right here. Mom may pass on green, so that's a possibility. Or it could be blue and yellow, okay? Blue and yellow, okay? So those are the possible combinations. So the half that gets passed on, so let's talk about this terminology. The half that gets passed on from each parent is called a haplotype, okay? All a haplotype is a group of closely linked alleles on a single chromosome, okay? So in the case of MHC, the genes that produce these molecules are so close to each other that they usually get passed on as a complete package 99% of the time. So all of this usually gets passed on together. So you can see all these alleles get passed on as a whole, okay? All these alleles get passed on as a whole, okay? They usually go over as a full package, okay? 99% of the time. But the other 1% of the time, something known as a recombination can happen. That just means instead of the entire chromosome being inherited, okay, some of dad's chromosome, A, let's say, will be inherited, and some of B will be inherited, okay? So instead of, let's say, A1, CW7, B8, and DR3, DQ2 all getting passed on together, maybe only 4 of those get passed on, and the 5th comes here, from here, okay? That's known as a recombination, okay? It's rare, but you need to be aware of it. But like I said, most of the time, the entire half moves over as a complete package. So 1/2 here, that makes up a haplotype, okay? So the 2 haplotypes inherited make up the complete set of MHC genes the child will carry. This is known as a genotype, okay? So those 2 halves come together. That's your complete picture. That's known as a genotype, when the 2 halves come together. The phenotype is what's actually expressed or observable. So if we go back to the hair color example, if the child inherits a brown color gene from dad and a blonde color one from mom, but the child actually has brown hair, okay? The genotype is brown blonde. That's the complete set of genes inherited. They got a brown from dad and a blonde from mom, okay? The genes are there, they're inherited, they're passed on. So the genotype is that complete set of genes, okay? So the genotype is brown blonde, but the phenotype is brown because the child has brown hair. That's what's actually expressed, okay? Because the dark colors are usually dominant, okay? So that's the phenotype. It's what's expressed. But MHC genes, like we mentioned before, are co-dominant. And that means everything inherited is expressed on the cell surfaces, okay? So all this, let's say this is the combination the child inherits. This is one haplotype, second haplotype. This whole thing, this whole set is known as a genotype, okay? The phenotype is what's actually going to be expressed. So because they're codominant, A1, A3 will both be expressed on the cells. C7, C8 will be expressed on the cells. B8, B14 will be both expressed on the cells. Everything is expressed together on the cell surfaces, okay? So in this case, the genotype is the same as the phenotype. Okay, so why is it important to know about all of this as a med tech? Because the answer is because there's some important testing applications. Okay, so one of the clinical applications is paternity testing. So, for example, determining a patient's MHC profile or tissue type, when used to determine paternity, what happens there is the lab collects samples from each of the family members and tests their white cells against a panel of known reagent antibodies to see where a reaction occurs. So if you want to know if the patient has, let's say, HLA-A2 on their cell surface, then what you do is you'd apply reagent antibody anti-HLA-A2 to the cell, okay? If there's a reaction, then you know that the patient has that particular antigen. So we'll get into some of the methods more specifically when we talk about pre-transplant testing in later lectures. But for now, just understand that based on this testing, the lab can determine a patient's tissue type. So in this example, the lab determines the tissue type of each family member to be as follows, as you see here on the screen, okay? So the mother's got A2, A3, B8, B12, C1, and C2. The child has A3, A9, B8, B16, C2, C1, and the father has A1, A9, B7, B14, C2, and C7. So how do we use this information to figure out if the father is in fact the child's father? Okay, so let's go through this. So knowing that half of the child's genetic material, MHC profile, comes from the mother, and let's start there. Okay, so look at what the child has: A3, A9. Okay, the mom has A3. Okay, so the child got A3 from the mom. The child has B8 and B16, so the child got B8 from mom. Okay, the child has C2 and C1, so so does the mother. So one of those came from the mother. All right. Now, whatever the child got from the mother, the balance should come from the father, right? So in this case, if the child got A3 from the mom, okay, the A9 should come from the father. Does the father have A9? Yes. The child got B8 from mom. Child also has B16. So B16 should come from the father. Does the father have B16? Oops, look at that. Nope. Okay. The father has B7 and B14. Neither of that matches up with the B16 here. Okay. The child has C2 and C1. Okay. So the child could have gotten C2 from the father and C1 from the mother. Okay. But the real discrepancy is right here. B8 came from mom. Okay. So B16 should come from dad, but dad doesn't have B16. Okay. So this child, this is not the child's father. So this is an important application of MHC antigen testing or tissue typing. Okay, so another application, as mentioned before, is in transplantation testing. Okay, so while MHC is super important for our immune systems and our survival as a species, the fact that it's so genetically polymorphic creates major obstacles in transplantation. Okay, we said that a patient's tissue type is almost like a fingerprint and virtually impossible to exactly duplicate outside of the immediate family. So that's why in organ transplants, it's impossible to go for an exact match, unless, of course, the donor is a member of the immediate family, in which case the chances become better. Okay. So depending on the organ being transplanted, there are different protocols as far as the minimum number of matching antigens required to proceed. Okay. A kidney is a good example. So current protocols require a match of 6 MHC antigens you see here. Okay. It doesn't have to be all of them because it's virtually impossible. So 6 is what the accepted protocol is. So as long as there's a match of 2 HLA-DRs, 2 HLA-Bs, 2 HLA-As, then you're good. Okay, then they can proceed. Okay, but what do you do about the ones that don't match? And that's the issue with transplantation. The recipient's immune system will recognize those as foreign and mount an immune response. So what happens is the recipient's or the host's T cells will connect to the donor's MHC1 or MHC2 molecules and recognize them as foreign. So cytotoxic T cells from the host will connect to MHC1 and destroy the donor tissue by direct attack, okay, through perforins and granzymes, like we already know about. And in the case of T helper cells, they'll connect to MHC2 and initiate destruction by indirect means, antibody production. So those antibodies, once they're produced, they will attack the tissue cells, okay? So to suppress that, patients are given specialized drugs called immunosuppressants. So we'll talk more about that in later lectures. But for now, just understand that HLA testing, tissue typing is very much applicable when you're talking about organ transplantation. Okay, so that's pretty much it. But to close this out, just click the button that you see on the screen to complete the final activity that summarizes the essential information covered in this lecture. So I can see where you guys stand with it, and it'll give me an idea of where you guys need clarification, if any. And I'll come back to you with more information. If you guys have any questions, if anything was unclear, then by all means log on to the chat session in Moodle, and I'll be there between 10 and 12 to answer any of your questions. Okay, so don't hesitate. See you guys soon.